In this course, you will learn what normalization is and how it works using 3 different strategies so that you can evaluate and determine the best normalization approach for your experimental conditions and context, as well as what types of variation normalization can correct for. Finally, we will walk you through the mechanics of how to apply normalization to your data sets. Let's start with the term normalization. Western blot normalization is a mathematical correction for small and unavoidable sample-to-sample and lane-to-lane variation that is computed by comparing an internal loading control to the protein of interest. The internal loading control acts as an indicator of sample protein concentration and is the tool that allows you to make relative comparisons of protein levels across your blot. Because the internal loading control is so important for accurate normalization, it must include all of the following characteristics. An internal loading control must be an endogenous protein or proteins present in all samples. The expression must not be affected by experimental parameters such as drug treatment, developmental stage, disease state, etc. And it must be detected within the same linear range as your protein of interest. For accurate normalization, the core principle of normalization must be valid. This principle states that the signals for the target protein and internal loading control must vary to the same degree with sample loading. This means that the target protein and internal loading control must be dependent on and be proportional to the sample protein concentration. The signals will then go up and down together as the protein concentration changes, giving you a consistent target to control ratio in all experimental samples. The core principle also tells us 2 important attributes of an accurate, effective internal loading control. First, the signal intensity of that loading control must be proportional to the sample protein concentration, and secondly, it must be proportional to the abundance of the loading control. These characteristics must be true for the internal loading control to stand in as our indicator of sample protein concentration. Otherwise, an internal loading control will not accurately correct for variability and can even introduce variability and noise during data analysis, making it harder to detect changes in your samples.